Dr. Thomas Grothues. He is a research associate professor at Rutgers University and the research coordinator of the Jacques Cousteau National Estuarine Research Reserve. His research focuses on how physical changes in environments impact the species composition and abundance of fishes. And he's worked on a wide range of species, from thresher sharks to horseshoe crabs to larval fish. And today he's going to talk about long-term observations of larval fish assemblages in the Mid-Atlantic.
Okay, thanks. It's actually a little bit different than the title I have, but I will be talking about larvae in here. And so let me start my slide sharing now. And thank you, Christina. And my hope is that you can see that. Yeah.
Looks good.
Okay, good. Cool. So this is where I am right now, and actually where Christina used to work, right here at the Rutgers University Marine Field Station in New Jersey. And this is looking right in the inlet. Just to the right and left of the screen are the tips of two barrier island systems through which the water flows to enter this system. So I'll get into a little bit more about that. I wanted to acknowledge that this work actually, much of it precedes me. Ken Abel, who was the director of the field station, started the long-term time series that I'm going to be taking data from long before I was even at the station. He has retired, and so he still comes here once in a while, but that's him right there. But over some 32 years now, this sampling program has continued, and I've started mining it together with Ken and Jason Morrison lately for questions about patterns that interest me. So a lot of people over the years have contributed to this, and it is a very labor-intensive work. I do have this this space saved for galleries, either Zoom or Teams or whatever. I'm going to try not to intrude in that space over there. This is more than just a Teams setup here. So, but to get back into the meat of the question here, the overview is we're going to talk about why I study larval fish in the first place. I want to introduce the study area, talk about the study question, which is how has the fish community here changed, both in latent sense and also in response to environmental factors. And while that is a larger question, today I'll be focusing, besides latent factors, just on temperature. And the data sets that I'm using are environmental loggers and plankton net collections. So I tried to make the average fish here. And the reason why is that when I present this kind of material to fishers in the community here, they frequently want to know why I study larval fish instead of like real fish or average fish, which here means like striped bass or bluefish. And so I drew the average fish I tried, and I decided immediately that it was a hamulid instead of an average fish. But even so, I think the only thing we can say about average fish really is that they're dead, because they start dying as soon as they are born or hatched. And that mortality rate exceeds 99%. And what that means is that the 10th to 100th% of survivorship changes a lot in terms of how many fish there are. It's not the mortality, but rather the survivorship, the inverse of the mortality, that really matters. So a very, very small change in mortality can result in a huge, huge change in survivorship because of the percentages that you're talking about. So a 0.1% survivorship versus a 0.2% survivorship is a doubling. Think that way. So we're going to look at what is happening during early life history for marine fishes. And this is typical of marine fishes worldwide, but in New Jersey here we have actually somewhat of an exaggerated life history segregation because of our seasonal seas and the fact that many of the fish here are estuarine dependent. And so I'm taking here a model of summer flounder, and summer flounder through spring and summer are in the estuary where they're growing, but as fall comes, they migrate out onto the continental shelf of the ocean, and they migrate all the way to the edge of the continental shelf. And during that time, they are spawning, and the spawn is pelagic drifting eggs that are fertilized. So they're broadcast spawn, and they last for a few days to weeks, depending on the water temperature, before they hatch and turn into larvae, which are still largely planktonic. At earlier stage, those larvae are friction bound and carried by currents. They do develop some ability to move up and down and change the direction they're going by selective tidal stream transport and selection of water masses, and eventually, as they're growing, they do enter the estuary, and then they metamorphose. That metamorphosis is pretty dramatic, and at that point they settle down to the substrate in the estuary, where they grow very, very quickly. So quickly, in fact, that some of the males are already sexually mature at the end of their first year, and at that point they join the migration before the winter freeze happens. So this is cyclical, and it's dependent and cued by temperature and potentially other things, such as storms that happen on a seasonal cycle. And such kind of cued events or life history stages is what we refer to as phenology. So it's periodic biological phenomenon correlated with climate conditions. And many species here and worldwide follow this kind of pelagic larval development and nursery before settlement, or even if it's not settlement, before recruitment to some kind of adult habitat. So what happens is that the cues over time can change. We expect them to change because we've already documented that there is change in many climate cues or seasonal cues here. And this being a particularly seasonal system, we kind of expect that to happen if we're not looking for it. We've got blinders on. So that is something that I am interested in. Okay, looking at sea surface temperature anomaly, we can see that the region here in the Mid-Atlantic Bight is particularly hard hit. We're warming very, very fast here for a number of different reasons, but it's certainly an area that needs attention in terms of climate change and temperature change specifically. And this is from the website where you can watch that movie run. It's just a still shot here. Where I'm doing this, so this is a map of New Jersey, and down here is the Jacques Cousteau National Estuarine Research Reserve, where I'm stationed. This is within the Mullica River watershed, and this entire watershed is a region that's preserved as part of the Pinelands National Preserve. It's a massive area, not what people think of as New Jersey, rather undeveloped. As I was saying earlier to the graduate student group, just an indicator of how rural it is. There's 300 registered fur trappers in the county. And so and what does happen in it is, in terms of agriculture, is blueberry and cranberry, which are native, so they're not fertilizer or pesticide intensive agriculture. Generally, this is maybe one of the cleanest systems along the Atlantic Northeast, and maybe even the Atlantic coast, as far as river systems goes, undammed and well protected. Additionally, it has infrastructure in it from Jacques National Estuarine Research Reserve. So I'm going to mention the loggers that are used there. And here's one of those. So the system-wide monitoring protocol is a system used by 29 reserves around the country, from Alaska down to the border of Mexico, Gulf Coast, up this side, Puerto Rico, and Hawaii as well. There's 4 sites. I'm going to be talking about data that comes from one of them down just off the Marine Field Station there. So here's the situation. We have among the highest annual temperature cycle of any oceanographic province worldwide. So these are daily mean temperatures. The tide can bring fluctuations of as much as 6 degrees in a single tide. That really that's really hidden by this mean temperature curve. So what happens here is that we get temperature from about negative 1.2 at the lowest to temperatures as high as 35 or 36 degrees C in summer. And this is the average annual temperature from, I think, from 16 years put together and smoothed. So it's a very seasonal system. Fish come in as recruits or as adult fish that spawn in the area and leave progeny behind. They migrate back out, and the larvae themselves also grow into larger fish that leave. We do get recruits of tropicals up here. We see typical Florida fish. We see trumpet fish, coronet fish, butterfly fishes, groupers and stuff. They recruit and then they die. If they can't make it out, we get a number of jacks, for instance, that can make it out. The system has been changing. This is using just data from the logger only, and we see this incredible change of 3 degrees over 19 years, which is, I would say, alarming. It is leveraged by a period here in 2012 that had particularly high temperature. If we look at the longer term record using data from back as far as 1990 that gets measured before the loggers were in, we see a little bit lower of a curve, and we also see some modes here that we went through: a low mode, a mode of high temperature, and a load of low temperature before going up and down again. On the whole, the temperature has been going up over this longer span at 1.2 degrees per year. So it's still going up, but the R squared value is much sloppier around that, and you can see why. And this becomes important later on when I talk about the decoupling of latent trends and temperature trends in the fish that I'm looking at. So I already mentioned that we see fish here from the south and from all these regions, but we also see fish here from the north. As far as just illustrating how much this changes, we have seen in this system in New Jersey, we have seen manatees, adult manatees and adult tarpon, but we've also seen beluga whales, so white whale has come down and visited, and we get cod and pollock and stuff like that, and haddock in winter. Over time, from previously published work, we've seen a general decrease in the incidence of fish larvae from northern species and a general increase in the larvae of fish from southern species. Both in richness and in the abundance of the actual numbers of those fish that are coming in. So that certainly piqued my interest. And this for juvenile fishes caught in traps, which is a similar survey, but not one I'm going to talk too much about today. And so we looked at the assemblage change over the series of plankton that I'm going to be talking about, incoming plankton. And I know it's impossible to read the particular fish that contribute to this trend, but this is looking at the first principal component of a PCA of fish species. Only the most important of which are shown here. There's 75 or so species in the PCA data set, and you can see that it has changed over time, and rightly so. As this was going through review for publication, a reviewer asked, what about the fish that that are still there, that have not changed in having disappeared or appeared, being northern species that disappeared, or new species that have entered the system and contributed to the increased richness, or diversity for that matter. Have they changed in their life history locally? So that's a great question, and was the very obvious next one to go to, and that's what I'm going to spend the rest of this time on. So we call that phenology, and I introduced the term earlier already. It is generally I should say that there is a lot of previous studies on phenology. But they come from systems where it's easy to do that, especially plants. So there's a number of ways to look at phenology. You can look at phenophase or phenostatus, especially when you're talking about plants. When do plants bud? Then later on, when do the same plants flower? Later on, when do those same plants lose their leaves? You can look at that over space. So where does a cherry tree bloom in for instance, Washington, D.C. versus where it blooms in Vermont? Excuse me, the timing of when that happens. So it's across space. And of course, monitoring individuals at the individual level within a population. So you could do that for fish, but it gets very expensive. So that, for instance, you'd have to tag a fish and have a tag system that lasts long enough, an acoustic tag or something that you get to see the coming and going of that fish over years. Excuse me just for a second here. I'm going to lose my voice. Okay, try that again. Okay, so the question is larval fish arrival changing? And we can do that from one place. Expectation is because we've seen the warming water, we'd get earlier occurrence of spring spawned progeny. We get later occurrence of summer or fall spawned progeny. Longer persistence of spring or summer spawned progeny, with the idea that they like the warm water and they're around longer, and maybe their spawning phase lasts longer. And a shorter persistence of winter or northern cold affiliated progeny. Again, here is the system. This is where the Rutgers University Marine Field Station is, and this is a channel that runs through the Sheepshead Peninsula. So water comes in here and it flows very rapidly through here, as well as coming around here through the logger, and it makes a big circulation here. So that flow is well documented, and it's unidirectional with incoming tide. So we have set up along, we being Royal We, I was not involved at the beginning of this, but I'll stick with that because it's easy. So set up a plankton sampling net that uses that flow. And so what I'm going to do is query that long-term data set, see if landmarks are reached earlier or later in terms of fish ingress as a latent trend, meaning uncorrelated with anything. See if there's a pattern to that landmark change, if there is one, and see if it correlates to temperature, and do that for multiple species. So this always happens at night. This is the net just prior to deployment at a bridge that goes over that that fast flowing and deep creek. That creek is not like it's a salt marsh creek, so it's about 15-20 feet deep. And the water flows at about 4 knots. So we have this 1 meter hoop mesh net. It has a flow meter inside it. There's another flow meter that is deployed next to it, so it can measure actual flow and anything that's happening in the net that's a net artifact. It's always deployed at night so that fish don't see it. There it's set as 3 half-hour sets, but those are treated as pseudo replicates. So we just put all the fish together and treat them as a single 1.5 hour tow, but clear the net every half hour. We, of course, quantify the flow and express the data as catch per unit effort. So that happens weekly, and it's been happening weekly since 1994, although it got standardized in 1995. Only a couple of weeks have been missed. Hurricane Sandy was one, where the road was washed out and the station was washed out and closed. And another was when the ice was too thick to break through to actually set the net. We had really fast ice in a couple of years. So this is an image taken during the daytime just to show that happening. So here is the primary net that's has a marker on it, and you can see that the flow is really moving. This is incoming tide water now, and there's a second line for a secondary flow meter. So we have the inside and outside flow meter, and we can tell, for instance, if the internal flow meter has jammed, and if it has, we'll use a correction from the external flow meter. Some of the things that we catch, these are American eel larvae. We get different angrollids and clupeids. Bunker over here are Atlantic menhaden. Flatfishes, so this is Sculpthalmus aquosus, the window pane, summer flounder, goby, and here's a Atlantic croaker. And they're, like I said, very seasonal. So the analysis here is that, and I've written this as pseudocode, just kind of popped the comments out of MATLAB code. So you see a loop here for each species. We look at, we basically find the threshold of, we define the threshold of 5% of the total in this case. And then for each year, we calculate the annual total. Then we look at that annual total and decide what is 10%, 25, 50, and 85% of that annual total and set that as the benchmark. Then we calculate the annual cumulative sum for each sample date. So as it adds up, we say on what sample date did we hit the 10th percentile of that annual, the 25th, the 50th, and the 85th percentile of that annual total. So the annual total is reset each year. So this is independent of the total size, supposedly. And we'll get into that. So yeah, total size, what happens? So here you have the Gaussian distribution with a general standard deviation equal to about the mean. And you can see where you have your 50th percentile and where you have, for instance, the 10th percentile of accumulation. So what happens if you have a smaller one? Your 50th percentile stays in the same place, assuming that the timing of everything else is the same. This is just a smaller population. The 10th percentile has now moved in the day of the year, even though even though the distribution around the mean has remained the same. So the 50th percentile should roughly be less vulnerable to that kind of effect, but actually is a Gaussian curve ever a reality? I think no, certainly not on the long term. So being as this hasn't been examined yet, we really needed some additional potential benchmarks. It's quite possible that these things are very skewed. And so that's why we set up this 10th 25th, 50th, 85th to look at sensitivity of benchmarks to being able to decide if something's happening. And we added a couple extra benchmarks. One is the timing, or I'll come back to that. So basically looking at two different curves here, you have an amplitude, so no fish come in and then a bunch come in and they have this very high amplitude peak and change to nothing again. And year two, that happens again with a smaller peak, fewer fish, but it's still at day 125. And so there's no change. Okay. And so we don't care about how many fish are coming in. Only thing we care about is if that has actually moved. So here you have the original mean at day 125, and here you have the mean occurrence, or the 50th percentile occurrence for year two. And so what we're going to do is take this and flip it on its side so that the day of the year is the y-axis. So now you have year one, you have those particular fish arriving at 125, and in year two at 135, day 135. And then we can do regressions on that. So we're back to that's what we did. And the caveat, though, that is if you take that day 1 through 366 or 365, you have to adjust that for winter recruits, because there's fish that recruit across winter, and it breaks that into a bimodal pattern for years. So rest assured that we just added a pseudo date to string that out so that the peak of those things would always fall towards the middle, so that you could see a shift to the left or the right instead of a bimodal distribution. So here is a quick look at a curve of fish coming in the black. This is the accumulation curve of those larval fish. This is where they hit the 10th percent benchmark, etc. I didn't show the 85th because I added that later, so the 85th would be somewhere around here, maybe day 110, 112 or so. And then actually we also looked at when they've hit their maximum daily CPUE, the highest catch per effort. When did that happen? And that could be at the very beginning or at the very end. And also then I added another variable, which is the time between the 10th and the 85th percentile. So even though the 85th is not drawn here, let's say it was here, it's the span between day 98 or whatever that is, or between 92 and day 112, and to see if that span is narrowing or broadening. So then you take those dates and you do a linear regression on them here. So if we have a slope up or down, that means it's actually it's coming. If the slope is up, that means that the date is getting later, okay, in the year. A downward slope means that you're seeing recruitment or entry earlier in the year because the date is getting lower over time. R squared, of course, is how tight the fit is, and whether we trust it is generally 0.5. Yes, we trust it a lot. 0.1, we do. Less than that or greater than that, not so much. And that is, I think, the way that that publishing culture has been for a while. I actually stand with Dushoff et al. 19, or I think it was 2019. That the use of these, especially when you're looking at these large data sets, we no longer want to use this is significant, and at 0.06 it wasn't significant, or even at 0.1, but rather to say what it did. It went up, it went down, we're clear on it, we're not clear on it, and to publish along with that a measure of how much we trust it. So yeah, it's going up, it's going down, and we trust it to this extent as shown by the P. So I'm going to be using that P value as a measure of trust rather than absolute yes, it did happen, or no, it didn't happen. And this reflects a trend in what the statisticians have been saying that we should be doing, and I agree with them. So looking at the 69 species that were in this data set, 21 of them actually turned out 20. I noticed too late that lady crab were in here at only 2%, but 20 of them had were represented in over 5% of the samples, and we used them. And they really go over a wide range of species and life history. So Atlantic menhaden or bunker, blue crab. So these fish kind of spawn everywhere all the time offshore. They spawn a little bit in the estuary too. We're going to assume that most of the ones that we see are from local spawning because the abundance should be higher from them. Blue crab is a species that moves out to from the estuary to the bay mouth, broadcast spawns. The larvae go out into the ocean. They make a loop and come back, some to this estuary, some to the local, so it's decoupled a little bit. This is a species that actually nests within the estuary. Summer flounder, one that winter spawns out on the shelf. Winter flounder, winter spawns, but inside the estuary, and a bunch of different kinds of habits and habitats are represented here. Second again. So here's an example of the ingress patterns that we see, and you can see how irregular they are, even to the point where there's some where you wonder if they spawned at all some years. But from 1990, when I said that wrong earlier, I said 95, but 1990 it started in 89 and became standardized in 1990. So you see some very high peaks in recruitment, some very low ones. On this particular scale, we can't really see where that's happening within the year, though we can see broad and narrow recruitment. Over here, we can see a really strong decrease in the day of the year of the benchmark, and this one happens to be for the 50th percentile benchmark. Here it is for American eel. So American eel, they spawn in the Sargasso Sea, and the larvae are transported by the Gulf Stream and queue in on freshwater plumes issuing from estuaries, and the elvers make their way up those plumes and seek out the estuaries and then move up from them as they as they mature. So you can see here a trend. If that's linear, it's very strong. It does tend to be a little bit nonlinear, but it's really a very, very strong change in when we're seeing the 50th percentile of ingress for American eel. But so unlike the Anchoa hepsetus, striped anchovy that spawn right off the bay mouth, these things are coming from a long ways away, and it's almost a year between when they were spawned and when they're entering the estuary, and there's a lot of stuff that happens in the meantime. Can also see that the CPU has decreased quite a bit, and so we do know that the overall shape of that or the height of that might affect the curve if it's Gaussian. So let's look at the general patterns that we found here. The position and color of the bubble. So anything on the left side of the graph that's in red, left side of 0, I should say, means that recruitment or entry is earlier. Anything that's blue or on the positive side of the graph means that entry or accumulation is later in the year. and we can see how much we trust that by how big the bubble is. So the bubble here is inverse of the p-value. So a very high p-value, in this case like 0.0000 something, probably 6 zeros I think before it got to be anything. Very highly trust that, and that happens to be for American eel. I think that was, yeah, that was for American eel. But in any case, we see the pattern for the different benchmarks. We can see something about the distribution in general. One thing that we see is that there's no real conflicts. We never saw the case where a particular benchmark, for instance, at 10% told us that it was going one way, and then at 25 or 50 or 85% that it was going the other way, for instance, later instead of earlier recruitment. There was no cases where all 6 of the benchmarks were trusted, significant, whatever you want to call that, at 0.1 or less of alpha. So, but we did have or for that matter, where all 5 benchmarks were that we trusted that all 5 benchmarks were changing. We did see several where at least 2 or 3 of the benchmarks indicated that there was change happening. And this is that distribution. In general, the 85th percentile was the most sensitive. We saw in most cases where that indicated at a high level of trust that benchmark was changing over time. Additionally, that with lower level of trust that's changing also. And then this is so that's for later, and this is for earlier. But in general, the 85th percentile benchmark did the best, and we're going to be using that benchmark when we move ahead and talk about the relationship between change and temperature. And you can see that we had some change in spread as well. So one of the questions that I keep harping on was if those things actually have normal Gaussian distributions. So the question that we can frame about that, because we look at it and we see that it's not, or actually very, very rarely the case, and in fact might change between years, whether it approaches a Gaussian distribution or whether it's skewed or kurtotic. So we look at P, or the trust that we have in a particular pattern as an evaluator or as a response to if that that ingress curve was at all flattened or skewed. And we find that there's a single case for the 50th percentile benchmark where we can actually detect with some confidence that the value of P, or the confidence that we have in the change, has to do with the shape of the curve, either in terms of the skew of the curve or the kurtosis of the curve. So for those of you unfamiliar with that term, kurtosis is the flattening. So imagine it's still kind of Gaussian, but it's flat like a plateau. And skew is whether the mode is and the tail is to one side or the other. So what does it mean? So we're going to look at temperature next, and what does it mean to have a negative correlation with a mean temperature? So if the temperature is up and the date is down, so it's earlier, that means that there's a negative relationship. So you can have a negative slope with temperature. That means in warmer years, the benchmark date is earlier. And this is what we get. And we did this only for the 85th percentile. We didn't just want to throw all the spaghetti on the wall and see what stuck. Having decided that there is at least an optimum way, or a best general way to do that. We stuck with it. So we're using the 85th percentile as the benchmark, and then looking at when that was reached relative to the temperature of a particular year. And we didn't do that across all the seasons. We tried to look at that only for the seasons that where the temperature is relevant to that species being in estuary. So we recognize that what's going on in the ocean is potentially quite different than what's happening in the estuary. For instance, measuring the preceding fall or winter for American eel, which are somewhere out in the Sargasso Sea or whatever, when they're not there yet, doesn't make much sense. So we looked at the ingress of these fish, when that timing was, if it was spring, summer, and we also kept the earlier timing, the preceding season, because that is when the adults of that species are spawning, and we've done that for those that are at least spawning locally. So, and then we got rid of the other ones, right? So here you can see in bold is the ones that pass that threshold of 0.1 or alpha of 0.1. I've left the other ones in there in compliance with what Dushoff is saying about reporting that. So you could see that, for instance, this just isn't even close. But basically, we do see a number of things that are changing with temperature, but it's kind of confused. So when we look at that pattern relative to the species that had latent change and life histories, they do not agree so well. So coming back to that question, is fish arrival timing changing with climate? The expectations were early occurrence of spring spawn progeny. There were some that did earlier. It wasn't, it was like 30–40%. Later occurrence of summer or fall spawn progeny, some, but there's also among the summer spawn progeny some that were earlier. Longer persistence of spring and summer spawn progeny, no, they got shorter. So maybe they're queuing on the change in temperature, and then it gets too hot, or it doesn't matter because they spawn in a batch. And shorter persistence of winter or northern progeny, no, except for eel. So there was longer persistence of one, but that was of eel, which is a winter spawned thing. The other ones were all shorter. So to wrap this up, the emergent trends, that the 85th percent benchmark is the most sensitive. The shape of the larval ingress curve is variable across species and within species across time. So this really, really complicates things. Later ingress was slightly favored over earlier ingress by number of species and by the strength of the relationship, both. And phenological shift for a slight majority of species correlated positively with temperature change in the estuary, but it didn't for other ones. Moreover, the species with clear temperature correlations were not necessarily the same ones as those that had clear latent shifts, meaning just that they just shifted over time. There was some that had clear temperature correlations that did not have clear latent trends, and there were also things with clear latent trends that did not have clear temperature correlations. So that went both ways. So, and we think the differences in the trends may relate to life history traits. However, the distribution of these trends relative to life history suggests that they're not random. There's a lot more we have to do with this work. I think the next step here, well, it begins with rampant speculation. So that's where I am here. I just wanted to show this image here as a way to back my rampant speculation. But hey, this is important because this is where we're going to form our hypotheses to move on with the work and be able to identify observations that we can test it with. But this shows the Gulf Stream here leaving Cape Hatteras at the Charleston Bump. You can see a number of features here. There's shingles. This is a sheared meander crest from the Gulf Stream. You see forward water here. So this is Mid-Atlantic Bight water, and on top of it you see fresher plume water, cool and warmer from Chesapeake Bay, coming together and being entrained through friction onto the side of the Gulf Stream, which is so warm that it even overruns that fresh water and leaves you with a freshwater lens underneath that I can't show here. And you have a warm core ring that is spiraling back, and from past ones you see that degrades and forms a slope C, and all these things are highly dynamic and influence what is brought to the estuaries along the coast here, and how those fish recruit. So there's a lot of so this also of course influences adult movement and the timing of adult movement for things like bluefish, weak fish, and stuff like that that make runs up and down the coast during their spawning season. So the adults change the progeny placement and potentially the timing, and that interacts with all this oceanography to give us our pattern. Overall, there is evidence of changing phenology at the examined temporal scale. It's moderate. I kind of thought I might have expected more in the sense the proportion of species is low, but the trend was very strong for individual species. There are numerous other phenological benchmarks to test, and some of these, for instance, would allow us to test against the specific types of oceanographic events that I mentioned. So again, coming back to the idea that the shape of the curve really matters in how the benchmark is assessed. We have we can look at dispersion, peak to peak amplitude, slew, rise time, pulse widths, etc., and use wavelet analysis in bringing that to oceanographic markers. We do have another long-term data set. I'll just do this in closing here. We have trap data. So traps look at settled fish. So it's still young fish, but this is, for instance, where we see groupers and allostomids and stuff like that. And it's much more tied to the temperature within the bay, because the fish that recruit to these traps are recruiting throughout the period that that they are here. And in fact, it's possible that a single individual actually recruits several times. So there's a matter of persistence here that's that gives you a different kind of persistence than the persistence that you measure when you measure span between larvae first coming into the estuary and disappearing, because those are generally the same age of larvae that are pulsing through, and that's very different with these traps. So we want to get into analyzing using similar methodology the trap data here, which is going to have a much more local signal. And that's going to be catching things like these. So there's gag, which is a southern species, and black sea bass. And I think that's good, given that this is a a video. We can't stare at Zoom forever, so I'm going to try to cut this one short in terms of me talking, and leave it to questions here. If anybody's still there. Thank you, Josh. Thank you. So I can go back to the slideshow to cover parts, but yeah. Go ahead.
Yeah, This is Chuan Hu. I know nothing about the fish, but I'm
Okay.
curious to know. You focused on temperature. What about the food availability, phytoplankton?
Right. Yep. That's a great question. So I think that will especially become a question when we talk about the trap data that I talked about at the very end as one of the next steps. So the fish that we're seeing in the plankton are there for a number of days sometimes before they're settling, right? They have it's really a fairly short. Now for instance, a fish species might be there for weeks. You might be catching that fish for many weeks, but the individuals themselves, once they enter the estuary, are transforming fast and settling. Some of them, of course, are coming in with egg yolks and are feeding. They're licithotrophic, so they're coming in and they're not feeding yet, and this is their first period of feeding, while other ones are ready to feed on plankton. We do have some nutrient data and phytoplankton data, but on a much shorter term. We don't have it going back to 1990. I think it's appropriate to use on a shorter data set for the trapped fishes, because we have a clear understanding that they're feeding locally and that their survival is tied to what's being measured here in the estuary instead of somewhere else out in the ocean. There have been studies looking at the general biomass and assemblage of fishes in this estuary and others, where rainfall, which has previously been correlated with nutrient level in the estuary, significantly increases the amount of fish and the richness of fish that you have in the estuary. And we think that's a trophic thing. So it's a great question. I haven't been able to get at it. I think it's very difficult to get at for larvae, but better for juvenile fish.
Thank you. Thank you.
You're welcome. Great question.
Yeah, the data, of course, it's a very precious data. Maybe you don't have enough data for phytoplankton. I understand the difficulty. I ask because many years ago, Trevor Platt published a paper. Every year there's a spring bloom, and depending on the timing of the bloom, the larvae survival rate is different.
Yes, so we've actually put in a proposal to the long-term ecosystem for the Northeast to do that kind of work for things that you can measure broadly. So the phytoplankton blooms, of course, are patchy, but they occur over a wide area. So it's appropriate to take wide area data, like for instance, recruitment of herring across the whole shelf, and look for correlations with phytoplankton blooms across the whole shelf, right? Because they're moving a lot. Much more difficult to do in the estuary, of course, with very recent fish. But it's a great question. I think it's really important. We have a very weak phytoplankton bloom here, in particular because the zooplankton aren't completely shut down in winter. And so they start to precede the bloom a little bit. They don't have to catch up the way that they have to in other shelf systems worldwide. And they start nipping the beginning of that bloom in the bud as it happens. And so it's not as remarkable as elsewhere.
Interesting. Thank you.
You're welcome. Any other questions? It's too close to happy hour, huh?
Well, if nobody else has questions,
go for
it. I have a simple one. So you have weekly sampling using net. So now, did you happen to catch any jellyfish?
Yes, and that's a great question too. So in fact, for about 5 years, we tabulated the jellies, right? And we have that as a short term, but they're really problematic. So we're you notice that this data went only to 2015. We're just now getting to actually identifying the animals from 2016 and 2017 because we pull up, you know, thousands, and they all have to be identified individually under a microscope. And when we have funding for that, we can do a push, and then when we don't, it just lags, right? So a consequence of that is that we immediately pull the fish from the plankton sample that night when they're still alive. You know, that I showed the picture, it comes in, and Christina used to do this with us. The sample gets put into a glass dish, and the larvae get pulled out and put into alcohol, right? And so then you have these little small glass vials, huge collections of them. We can't keep the jellies, right? It's years later, and you can't pickle them. When you put them in alcohol, they just dissolve. So actually quantifying jellies was a push for 4 years when we had funding specifically for that. There's another student, that was her dissertation, so I'm not very familiar with that work, but they do eat larval fish. There's really great publications on how they can absolutely slam larval fish, and if their phenology coincides with the phenology of the larvae, they could change the shape of that curve and make the benchmark appear much earlier, even if the total amount is not important.
Thank you. Very, very interesting.
Thanks for the great question.
All right. Tracy, go ahead.
Figure out how to turn myself back on here.
Hello, Tracy.
Hi. So I've done some work with a similar, a large-scale phenological study with euphausids in the off of Oregon, and one, we found that the jellyfish will eat all the krill eggs, but two, we also found that that the spring blooms are really closely correlated with the timing of the start of the upwelling season. And I wondered if there's a similar sort of environmental parameter that might correlate with the spring bloom where you are.
Yes, for sure. So we can kind of figure out when the spring bloom is. It's just not as pronounced, but it happens. And there are some other important indicators. And when we do that, such as the formation of the cold pool, for instance, but we can do that for very specific species, right? Because we have to dig down into the life history then. So I think, for instance, what happens for mummy chog is going to be something very different than happens for seahorse and Atlantic herring, which of course Atlantic herring would be much more tied to the coastal shelf system, and so would croaker, but they would be tied to what's going on to the south because croakers are much more southern fish and they come up. One of the real interesting things we noticed was that croaker did not have a strong relationship here with temperature, but we know that they're absolutely limited by temperature through another aspect. So croaker are at the northern edge of their distribution here. They are moving up along the coast in fall. They're spawning. We see lots and lots of croaker larvae. They can be the dominant species October, November, and then what happens if you sample the young with a trawl? If it was a warm winter, juvenile croakers that have overwintered as larvae or settled juveniles in the system are the dominant fish in spring in the assemblage, at least the benthic assemblage. And if it was a cold winter, there's none. It's just a threshold dynamic that is set by some winter temperature threshold of survival. So it has very little correlation with what's happening out. They seem really tolerant as adults of where they're spawning and where they're putting their progeny, which is kind of everywhere. And then survival of the juveniles is really what matters.
Yeah.
That's work published by Hare and Abel, 2007, I think.
Okay. Thank you.
Thank you.
Are there any other questions? Tracy, your hand is still up.
Or again, that's fine too.
Or she can ask again, of course. Okay, if there are no more questions, then let's give a great round of applause for a fantastic talk. Thank you so much for
Thank you.
a great talk and being so willing to change the time.
No